Hello everyone. Today we are going to discuss about polymerases. As we all know, the basic activity of polymerases is formation of phosphodiester bonds. Introduction to polymerases. These are the enzymes that catalyze the polymerization of nucleotides into nucleic acids, that is to DNA and RNA. Polymerases play a crucial role in DNA replication, DNA repair and gene expression. There are several types of polymerases and each with specific function. As we have discussed earlier, these are the enzymes that play a crucial role in synthesis of polymerases, particularly nucleic acids such as DNA and RNA. These enzymes are responsible for catalyzing the formation of phosphodiester bonds between nucleotides. These nucleotides are the building blocks of DNA and RNA molecules. There are different types of polymerases that are involved in DNA replication, DNA repair and gene expression. The various types are DNA polymerases, RNA polymerases, epsilon polymerases, beta polymerases, etc. Now let us discuss about the DNA polymerases. DNA polymerases are responsible for synthesizing DNA molecules from deoxyribonucleic acids. These require a template strand and a primer to initiate synthesis. High fidelity DNA polymerases such as Taq polymerase are commonly used in PCR. As we can see in the image, helicase unwinds the DNA and a leading strand and lagging strand are produced for the template strand. These DNA polymerases, the basic function of these is they add nucleotides to the growing DNA chain in a complementary manner using one strand of DNA as a template. They also play a role in DNA repair process. The three major types of DNA polymerases are DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. The basic function of DNA polymerase 1 is it removes RNA primers and fills the resulting gaps with DNA during the DNA replication and repair mechanism. That is, DNA polymerase 1 is basically involved in DNA repair mechanism. They remove the RNA primers from the repair site and add the DNA molecules. DNA polymerase 2 is involved in DNA repair mechanism. Coming to the DNA polymerase 3, this is the main enzyme that is responsible for DNA synthesis during replication in bacteria. The other types of DNA polymerases are DNA polymerase 4 and 5. These are also involved in DNA repair process, particularly when DNA is damaged. Coming to the RNA polymerase transcribe DNA into RNA molecules. These require a promoter sequence to initiate the transcription. There are three types of RNA polymerases, as in the case of DNA polymerase in eukaryotes. Each is responsible for transcribing a different class of RNA. RNA polymerases bind to specific sequence of a DNA called the promoter and they initiate the transcription process. This ultimately results in the formation of messenger RNA, transfer RNA and other types of RNA molecules. RNA polymerases are also of three types, 1, 2 and 3. RNA polymerase 1. This is involved in synthesis of ribosomal RNA. RNA polymerase 2. It is found in the nucleus and synthesizes the messenger RNA. RNA polymerase 3, which is also found in the nucleus and synthesizes transfer RNA and some other small RNA molecules. Coming to the reverse transcriptase, these are the enzymes that synthesize DNA from the RNA template. They are commonly used in techniques such as RT-PCR and cDNA synthesis. In case of HIV, they use the reverse transcriptase to convert their RNA genome into DNA, that is the retroviruses. The other type of polymerases is beta polymerases. Beta polymerases, these are involved in DNA repair. They can bypass damaged DNA base and insert the correct nucleotide. Deficiencies in beta polymerase have been linked to cancer and immunodeficiency disorders. The other type of polymerases is the epsilon polymerases. These epsilon polymerases are high fidelity DNA polymerases that are involved in DNA replication. They are essential for maintaining genome stability and preventing mutations. Mutations in epsilon polymerases have been linked to cancer and genetic disorders. Coming to the sigma factors are the protein that control the specificity of RNA polymerase binding to promoter sequence. These sigma factors allow RNA polymerase to recognize different classes of genes and regulate gene expression. Different sigma factors are expressed in response to environmental signals and developmental cause. Polymerase chain reaction (PCR). PCR is a molecular biology technique that amplifies DNA sequence in vitro. That is, the DNA sequence is amplified in vitro techniques. It requires a template DNA strand, primers, high fidelity DNA polymerase, and nucleotides. PCR has revolutionized molecular biology and has numerous applications in research, medicine, and forensics. Coming to the conclusion of polymerases, on a whole in this full PPT we have learned that polymerases are essential enzymes that play a crucial role in DNA replication, transcription, repair and gene expression. There are several types of polymerases, each with a specific function and mechanism of action. Advances in polymerases research have led to development of numerous molecular biology techniques that have transformed the field of biology. The enzymes involved in DNA replication are primerases, DNA polymerase 3, 1 and DNA ligase. As we all know, the function of this is they are involved in synthesis of RNA primer or synthesis of new DNA strand or replication or repair of DNA, etc. Thank you.